Hey there, curious minds. Welcome back to the deep dive. Today, we're going to peel back the layers on an organ we often take for granted, the lungs. You probably know them for helping you breathe, exchanging gases. But as we're about to uncover, they are truly multi-talented, acting as both, well, a vigilant fortress and a sophisticated chemical plant all at once. We've got some incredible source material today. It's a thorough exploration from a top-tier physiology text, really packed with fascinating details. Details on how your lungs don't just help you take in air, but also vigorously defend you against the outside world and meticulously manage your body's chemistry. Our mission is to walk you through the lungs' incredible, often invisible defense mechanisms, everything from tiny dust particles to complex viruses, and then explore their often overlooked metabolic superpowers. Get ready to truly understand this information, really dive deep into how these vital systems work.
Yeah, what's really fascinating here is that the lungs represent our body's primary barrier. I mean, they're constantly interacting with the environment with every single breath we take. Understanding their dual role in host defense and metabolism, it really reveals a level of biological sophistication that is, well, truly remarkable.
Okay, let's unpack this incredible frontline defense then, the mucociliary clearance system. We inhale so much stuff every day, dust, pollen, microbes, you name it. So how does this intricate system actually work? How does it remove all that foreign material?
Well, the core of this system involves three major components working together, kind of in concert. First, you have two fluid layers. There's the deeper sol layer, which is also called periciliary fluid. And then on top of that, you've got the gel layer, or mucus. These layers work with the cilia, and cilia are these tiny hair-like structures on the surface of the bronchial epithelial cells that line your airways.
Okay, so let's start with that deeper layer, the periciliary fluid. What is it actually made of, and how is its volume so precisely controlled? Sounds like a really delicate balance.
It is. It really is. It's a non-viscous, serous fluid, so basically thin and watery. It's produced by the cells lining your airways, these pseudostratified ciliated columnar epithelial cells. And these cells have this remarkable ability to either secrete fluid, that's largely managed by what are called CFTR chloride ion channels, or they can reabsorb fluid through epithelial sodium channels, known as ENC. This precise balance creates an osmotic gradient, basically, a difference in salt concentration, and that pulls water passively into or out of the layer. In a healthy lung, this layer is precisely 5 to 6 micrometers deep.
Wow, that's specific.
Yeah, think of it like a perfectly calibrated water level, because that's the optimal depth for those cilia to eat effectively. If it's too deep or too shallow, they can't work properly.
Right, and here's where it gets really interesting, I think, especially when this delicate balance is disrupted. We're talking about cystic fibrosis, CF.
Indeed. CFCF is the most common lethal inherited disease among Caucasians, and it's caused by mutations in that very CFTR gene we just mentioned. It leads to chronic bacterial lung infection and a progressive decline in lung function, unfortunately. For instance, there's one mutation, G551D CFTR, found in about 5% of patients. In this case, the CFTR chloride channel does actually reach the cell surface, but it just doesn't secrete chloride effectively. It's like a gate that's stuck closed. But the good news is precision medicines like ivacaftor, brand name Kalydeco, have been developed. They essentially stimulate that channel, kind of prop the gate open, significantly improving lung function and slowing disease progression for these specific patients. It's a fantastic example of targeted therapy.
That's amazing. And what about the more common mutation, the F508L CFTR?
Right, the F508del CFTR mutation. That affects about half of all CF patients, so it's much more prevalent. And it's different. Here, the CFTR chloride channel often doesn't even reach the cell membrane properly. It's misfolded and gets degraded before it even gets there. However, combination therapies, things like lumacaftor, ivacaftor, which is our combi, and tezacaftor, ivacaftor, or Simdego, and more recently, alexacaftor, tezacaftor, ivacaftor, that's Trikafta. These have really revolutionized treatment. These drugs work to correct this misfolding and transport defect. They help more functional CFTR get to the membrane and improve chloride transport.
So they're fixing the protein itself and helping it get to the right place.
Exactly. And these advancements have drastically altered the disease progression and, importantly, the quality of life for many, many patients.
It's truly remarkable how a single genetic error can unravel such a critical system, but also how these targeted therapies are now rewriting that story for patients. What a breakthrough. So, okay, with the periciliary fluid understood, let's move to the sticky part, the mucus layer. What's it like and who produces it?
Right, the mucus layer. It sits directly on top of the periciliary fluid, and it serves as a primary trap for inhaled substances. Think of it like flypaper. It's predominantly water, about 95% to 97%, which might surprise people.
Yeah, you think of mucus as thick and gloopy.
Well, it has unique properties. It's typically 5 to 10 micrometers thick.
Huh.
And it exists not as a continuous sheet, but more like a discontinuous blanket, almost like islands or patches of mucus. Its unique properties are low viscosity, so it's not too thick normally, and high elasticity, allowing it to stretch and recoil. And it's primarily composed of these large molecules called glycoproteins, or mucins. Healthy individuals produce roughly 100 milliliters of mucus daily.
Wow, okay. Enough to fill a small glass.
Pretty much.
So who are the cellular contributors to this mucus blanket? Where does it come from?
There are 4 main cell types that contribute. You have goblet cells, sometimes called surface secretory cells, and Clara cells. Both of those are found within the tracheobronchial epithelium itself, the lining. Then you have mucus cells and serous cells, which are located deeper within the tracheobronchial submucosal glands.
Okay, let's break this down. Goblet cells first.
Sure. Goblet cells typically make up about 15 to 20% of the tracheobronchial epithelium. They're found throughout the larger airways, generally up to about the 12th division or branching point of the tracheobronchial tree. But in many respiratory diseases like chronic bronchitis or asthma, they actually proliferate, they increase in number, and extend further down into smaller airways.
And that causes problems.
Yes, that can lead to mucus plugging and makes those smaller airways more susceptible to obstruction. They secrete neutral and acidic glycoproteins. And importantly, their mucus tends to become more viscous, thicker with injury or infection, which can worsen blockage.
Got it. And what about the submucosal glands? Where do they fit in?
The submucosal glands are present wherever cartilage exists in the upper airways, so in the trachea and larger bronchi, the more rigid parts. They secrete both serous or watery products and mucus products. Their function is quite dynamic. It's influenced by both sympathetic and parasympathetic nerve fibers.
Ah, so the nervous system controls mucus production too.
It does. For instance, cholinergic compounds, like acetylcholine from the parasympathetic system, stimulate secretion. Adrenergic compounds, like norepinephrine from the sympathetic system, can inhibit it. And local inflammatory mediators, things like histamine, released during allergic reactions, also stimulate mucus production. Within these glands, you have different cell types. Serous cells are often found more distally, further down the gland duct. They produce crucial antimicrobial agents like lysozyme and lactoferrin, while mucus cells are more in the middle to distal regions and contain these larger granules packed with mucus.
Okay, and we also heard about Clara cells earlier. What's their unique contribution?
Right, Clara cells. They're located deeper in the airways, in the bronchioles. They secrete a non-mucinous material, so it's not classic mucus, but it contains carbohydrates and proteins. This contributes to the overall composition of the airway lining fluid. And importantly, these cells also play a vital role in bronchial regeneration after injury. They help the airway heal itself.
Fascinating. Okay, now let's talk about sputum. We cough this stuff up sometimes. It's more than just mucus, right? I've always heard that its color can tell you a lot.
That's absolutely right. Sputum is expectorated mucus, sure, but it's really a mix. It also contains things like serum proteins, lipids, electrolytes, calcium ions, and even DNA from degenerated white blood cells, especially during infection. Plus, secretions from the nasal passages, the mouth, the pharynx, it all gets mixed together.
Yep.
And yes, its color can indeed be very informative for clinicians. It often correlates more with how long it's been sitting in the lower respiratory tract and the types of inflammatory cells or even red blood cells present, rather than just, say, the simple presence or absence of bacteria at that exact moment. It's like a diagnostic clue.
So what do the colors tell us? Any common signals we should know about?
Well, clear or maybe cloudy white, thin mucus is generally considered normal. But if you notice increased amounts or it gets thicker, that can signal an early infection or inflammation. Thick white mucus might indicate conditions like bronchiectasis, where the airways are damaged and widened, or cystic fibrosis, or sometimes even a lung abscess.
Okay, what about other colors? Red.
Red mucus suggests the presence of red blood cells, which is always concerning. It can be associated with serious conditions like pneumococcal pneumonia, lung cancer, tuberculosis, or pulmonary emboli, which are blood clots in the lungs. Needs investigation.
Definitely pink.
Pink mucus often points to the breakdown of eosinophils. These are a type of white blood cell very common in individuals with allergies or asthma.
The allergy connection and the less appealing colors, gray, brown, black.
Yeah, those are typically linked to inhaled irritants. Things like cigarette or marijuana smoke, sometimes cocaine use. Also air pollution from workplace environments, like coal mines leading to black lung. Or it could simply be old, degraded blood.
That's fascinating how a simple visual cue can reveal so much about what's happening inside the lungs. Finally, let's visualize the cilia, these tiny structures. How do they manage to move all that mucus, and what does their coordinated beat actually look like?
Right, the cilia, the respiratory tract all the way down to the bronchioles, is lined by this special pseudostratified ciliated columnar epithelium. Each one of those epithelial cells has approximately 250 cilia sticking out from its surface.
250 per cell. Wow.
Yeah, quite dense. And each cilium is just 2 to 5 micrometers long, incredibly tiny. They are complex structures themselves, composed of 9 microtubular doublets surrounding 2 central microtubules. It's a classic 9 plus 2 structure. And they have these things called dynein arms, which contain an ATPase that's a molecular motor responsible for their contractile beat. It uses energy to make them move. And they beat in this coordinated wave-like rhythm called metachronism.
Metachronism.
Yeah, think of it like a stadium wave going across the crowd. Each cilium moves slightly after the one next to it, creating this synchronized push. They beat incredibly fast, about 1,000 strokes per minute. During their power-forward stroke, they extend upward, fully into the viscous mucus layer and physically push it, along with any trapped particles, upwards or downwards, depending on location. Then, on the recovery stroke, they withdraw completely back into the thinner, watery sole layer, kind of tucking themselves away, ready for the next push.
So they only engage the mucus on the power stroke.
Clever. Exactly. Cilia in the nasopharynx propel mucus down towards the pharynx to be swallowed, whereas in the trachea and bronchi, they propel it upward towards the pharynx, again, usually for swallowing. Most of it we swallow without ever noticing.
An amazing self-cleaning system. So, okay, a lot gets inhaled and is immediately met by this amazing system. But what determines where these particles actually land in the lungs, and how are they eventually cleared if they manage to get past these initial defenses?
Right, so particle deposition, where things land, depends on several factors. The particle size is a big one, also its density, its shape, how far it has to travel through the airways, the speed of the airflow, and even the relative humidity of the air. There are basically 4 major mechanisms for this deposition, and each one is more relevant for different particle types or sizes. You've got impaction, sedimentation, interception, and Brownian movement.
Okay, can you break down each of those mechanisms for us? Let's start with impaction.
Absolutely. So particles that are larger than 10 micrometers. Think of large pollen grains or dust. They're mostly deposited by impaction, right up in your nasal passages. They're just too big and have too much inertia to make the sharp turns as the air flows through your nose.
They just crash into the walls.
Pretty much. So these rarely reach the lower respiratory tract. Now, particles between 2 to 10 micrometers, smaller dust, some bacteria, they're deposited predominantly by inertial impaction as well. But further down, in areas of turbulent airflow like the nasopharynx, the trachea, and the larger bronchi, especially where the airways bifurcate, where they split, their tendency to move in a straight line prevents them from changing directions quickly with the swirling airflow, so they impact there.
Okay, that makes sense. What about smaller particles? Sedimentation.
Right, smaller particles, say between 0.2 to 2 micrometers. This includes many bacteria and fine dust. These are deposited mainly by sedimentation in the more distal airways, the smaller bronchioles, where the airflow is much slower. Here, gravity simply has enough time to pull them down onto the airway surfaces.
And interception, that sounds a bit different.
It is. Interception is particularly relevant for elongated particles, like fibers, think asbestos or Selga fibers. Here, deposition happens when the particle's distal tip, its end, physically contacts a cell, or the mucus layer, even if its center of gravity might otherwise allow it to remain airborne and follow the airflow streamlines. Its shape causes it to get snagged.
Got it. And finally, Brownian movement for the tiniest ones.
Exactly. The tiniest particles, those smaller than 0.2 micrometers like viruses or ultrafine combustion particles, they are deposited in the smallest airways and right down in the alveoli, the air sacs, primarily by Brownian movement. This is basically random movement caused by collisions with gas molecules. It's influenced by their diffusion coefficient, and deposition actually gets more efficient as their size decreases in this range.
So the smallest ones move randomly and eventually just bump into a surface.
That's the idea. And unlike larger particles, density doesn't significantly influence the diffusion of these tiny ones. It's more of that random walk.
It's incredible how physics dictates where all these different inhaled things end up. So once deposited, how quickly are these particles cleared? Is it always a rapid process like that mucus escalator?
Well, in the conducting airways, the parts with cilia, the mucociliary system, is quite efficient, yes. It transports deposited particles from the terminal bronchioles all the way up to the major airways, where they can be coughed up and swallowed. This often happens within minutes to hours, but the clearance rates vary significantly depending on where you are. It can be pretty fast, maybe 5 to 20 micrometers per minute in the trachea and main bronchi. but it's much, much slower down in the smallest ciliated airways. The bronchioles may be only 0.5 to 1 micrometer per minute.
A huge difference.
A huge difference. And generally, the longer any inhaled material remains stuck in the airways, the greater the probability that it will cause lung damage. And this leads us to what's often called the Achilles heel of the whole lung clearance system. That's the region from the terminal bronchioles, the very end of the conducting airways, down into the alveoli themselves. This crucial area is completely devoid of ciliated cells. There's no escalator there.
Ah, so clearance must be different there.
Completely different. It relies almost entirely on alveolar macrophages, these immune cells that engulf particles. And that process is much, much slower. Days, weeks, even longer. And unfortunately, because particles can get stuck there with no quick way out, this region is the most common location for many occupational lung diseases.
Which brings us right to a relevant and sadly impactful clinical example, silicosis and pneumoconiosis. We've heard of black lung disease in coal miners. How does this Achilles heel connect to those conditions?
Yeah, these are classic tragic examples of diseases stemming from environmental workplace exposure. What happens is that those alveolar macrophages, our frontline immune cells down in the deep lung, they phagocytize, they eat or engulf the silica or coal dust particles. But crucially, these macrophages cannot destroy these inorganic particles. They just can't break them down. So the particles persist inside the macrophage, eventually leading to the macrophage's death and release of the particles, plus inflammatory signals. This whole process localizes and concentrates the particles right in that Achilles heel region of the lung, the alveoli and surrounding tissue where there's no mucociliary clearance to sweep them away.
They just accumulate.
They accumulate. And without that mechanical removal, these particles eventually enter the lung's interstitial tissue, the space between the air sacs. This triggers a chronic, unrelenting inflammatory response that leads to the formation of granulomatous lesions and widespread fibrosis, essentially, scarring. This scarring makes the lungs stiff and unable to expand properly, resulting in a restrictive lung disease.
Terrible consequences.
Terrible. Thankfully, increased awareness of the causes and much improved workplace safety standards have significantly reduced the incidence of these diseases, but they remain really important historical and clinical examples of how our defenses can be overwhelmed by chronic exposure.
Absolutely. Okay, so beyond the mechanical barrier, the lungs have this incredibly sophisticated immune system. How is it different from the general immune system we might be familiar with, say, the one operating in our blood or spleen?
That's a really crucial distinction to make. In non-mucosal tissues, like your spleen or liver, the primary defense is typically a classical, pro-inflammatory, antigen-specific adaptive immune response. This involves T lymphocytes, the ones with TCR T-cell receptors recognizing specific invaders, and plasma B cells synthesizing antibodies like IgM and IgG, which are potent inflammation activators. But mucosal tissues, and this includes your respiratory tract, your gut, your urinary system, even your eyes, nose, throat, mouth, lip, they're different. They're constantly interacting with the outside world. Billions of microbes, food particles, dust.
A constant barrage.
Exactly. So they've evolved to continually discriminate between what is harmful and what is harmless. Because inflammation, while protective, usually disrupts normal physiological processes. You don't want constant inflammation in your lungs or gut just because you breathed in some pollen or ate some food.
Right, that would be counterproductive.
Highly. So accordingly, mucosal tissues have developed specialized, non-inflammatory, or perhaps inflammation-sparing defense mechanisms. The mucosal immune system contains both specialized innate lymphoid cells, we'll talk more about those, like macrophages, natural killer cells, dendritic cells, alongside adaptive T lymphocytes that have a different type of receptor, the TCR T-cell receptors, and also plasma B cells that specialize in synthesizing IgA antibodies. These unique responses are geared towards preventing or limiting reactions to harmless foreign agents while still efficiently eliminating pathogens, ideally with minimal inflammation and tissue damage.
A more nuanced approach.
A much more nuanced approach. It's a frontline defense system designed to avoid unnecessary inflammatory responses. Of course, if these frontline defenses are bypassed or somehow fail, the lungs can still mount that classical adaptive immune response via the mediastinal lymph nodes, the ones located in the chest cavity. It acts as a backup system.
And these lymphoid aggregates play a unique role here, right? You mentioned BALT. We're not talking about typical lymph nodes like the ones that swell up in your neck.
Exactly. A distinctive feature of the mucosal immune system is antigen processing, but through these specialized lymphoid aggregates rather than true lymph nodes. Unlike lymph nodes, which have both afferent lymph flow in and efferent lymph flow out, these aggregates, like BALT, that's bronchus-associated lymphoid tissue in the lungs only have afferent drainage. Material flows in, but there's no defined exit vessel.
So it's more like a collection point.
Kind of like a sampling station or collection point, yes. BALT is present, scattered within the conducting airways. And an interesting feature of BALT is that the airway epithelium directly overlying it is non-ciliated. It's called a lymphopithelium. This creates a little break in the mucus flow, acting almost like a drain or a little eddy current to facilitate the collection and immune processing of foreign particulates that get swept towards it. It's a very targeted sampling system.
Clever design. So let's zoom in on some of these specialized adaptive lymphoid cells. Tell us about IgA. It sounds like it's specially designed for the mucosal environment, particularly the lungs.
It absolutely is. One of the key specialized features of all mucosal-associated lymphoid tissue, including BALT, is this unique antibody system centered on immunoglobulin A, or IgA. IgA has several specialized features that make it ideal for this environment. First, it's generally non-complement fixing. This means it doesn't typically trigger that strong, widespread inflammatory cascade that other antibodies like IgG or IgM can initiate.
Keeping things calm.
Exactly, keeping things relatively calm. It also has something called a J chain, which helps it link together and be transported across epithelial cells. And it usually exists as a dimer, two IgA molecules joined together, which gives it enhanced stability out there in the airway lumen, where it might face harsh conditions like enzymes. Plasma cells located in the submucosal areas beneath the epithelium synthesize and secrete this dimeric IgA. It then migrates up to the surface of the epithelial cell, binds to a special receptor called the polymeric immunoglobulin receptor, polyogic receptor, gets taken inside the cell, endocytosed, transported across the cell, and then secreted out into the airway lumen. And during the secretion process, a part of that receptor, called the secretory component or secretory piece, remains attached to the IgA dimer.
Why is that important?
It's crucial because it protects the IgA from being broken down by proteolytic enzymes that might be present in the mucus or secreted by bacteria. So this IgA antigen immune complex is highly effective at binding particulates, bacteria, viruses, essentially neutralizing them and clumping them together. This promotes their removal via the mucociliary clearance system before they can even invade the host cells, all without inducing excessive damaging inflammation. It's like a quiet, efficient cleanup crew.
Very elegant. And what about those other T cells you mentioned, the TCO T cells? They sound like a fascinating bridge between innate and adaptive immunity.
They truly are. They sit right at that interface.
Yeah.
While most of the classical adaptive T lymphocytes in our blood and lymph nodes are TCR T cells, TCR T cells also mature in the thymus, but the majority of them preferentially migrate out to mucosal tissues, the lungs, the intestines, the skin. They're a major T cell population there, even though they're a minority in peripheral blood.
What makes them bridge that gap?
What's unique is that they can recognize common molecular patterns, those PMPs, pathogen-associated molecular patterns, that are found on many different types of pathogens. Things like lipids or stress-induced molecules on host cells. This allows them to mount an immediate response, much like innate immune cells, without the need for classical antigen presentation by other cells, though they can also respond in that manner. So, because they express this duality responding quickly like innate cells but having some adaptive T-cell characteristics, they're considered a crucial link between the adaptive and innate immune systems. Furthermore, TCR T cells don't generally demonstrate classical immunological memory like OTT cells do, but they provide vital early protection without inducing excessive inflammation. They've been shown to expand rapidly in response to various viral and bacterial infections, and they can synthesize either pro-inflammatory cytokines to help fight infection or regulatory cytokines to dampen inflammation, adapting their response based on the situation. Importantly, they also seem to suppress IgE responses, the antibody type involved in allergies, which can help prevent allergen-induced inflammation in the airways.
Really versatile cells. That leads us perfectly into the broader category of specialized innate lymphoid cells, or ILCs. What sets them apart from other immune cells, and what makes them so advantageous in the lung?
Right, ILCs. This is a relatively newer classification, and it's a diverse group that functionally includes those TCR T cells we just discussed, but also dendritic cells, DCs, macrophages, and natural killer NK cells. What primarily sets them apart is that they lack antigen specificity in the classical sense. They don't have rearranged receptors recognizing unique epitopes like B and T cells do. Instead, they distinguish self from non-self primarily through the recognition of those conserved pathogen-associated molecular patterns, PMMPs, on pathogens.
Like the bacterial cell wall components we mentioned?
Exactly. PMPs are common identifying features shared by broad classes of microbes, things like peptidoglycans in bacterial cell walls, lipopolysaccharides or PS for endotoxin from gram-negative bacteria, or specific forms of viral RNA or DNA. Our host defense cells, including ILCs and even epithelial cells, have specific pattern recognition receptors, or PRRs. They're designed to bind to these PMPs. This binding triggers an immediate response.
Okay, tell us more about these PRs. How do they actually work?
Well, the two major classifications are toll-like receptors, or TLRs, which are mainly expressed on cell surfaces or within internal compartments called endosomes, and nucleotide oligomerization domain, NOD-like receptors, or NLRs, which are found inside the cell cytoplasm. Both of these receptor families, when they bind their specific PMP ligand, initiate intracellular signaling cascades. These pathways often converge on activating a crucial molecular switch inside the cell nucleus called NFKB, nuclear factor kappa B. NFK is a transcription factor that then turns on genes responsible for producing molecules that cause inflammation, like cytokines TNF-A, IL-1, IL-6, and fight viruses, like antiviral type B interferons.
And different TLRs recognize different threats.
Precisely. They have different specificities, acting like specialized sensors or keys. For example, TLR2 is particularly important for recognizing components of gram-positive bacteria. TLR4 is the main receptor for LPS, or endotoxin, from gram-negative bacteria. In the lung, various cells express these bronchial epithelial cells, alveolar epithelial cells, macrophages, dendritic cells. They all have TLRs. Other TLRs are specialized for detecting viruses. TLR3 binds to double-stranded RNA, a common feature of many viral replication cycles. TLR7 and TLR8 bind to single-stranded RNA found in other viruses.
So it's a sophisticated detection system. What's the big advantage of these ILCs having this system?
Perhaps the most intriguing aspect and the biggest advantage, especially in a place like the lungs, is their immediate response to pathogens. Contrast that with the days or even weeks it takes for a classical adaptive immune response to fully mobilize, involving clonal expansion of specific T and B cells and the generation of memory.
Speed is critical.
Speed is absolutely critical in mucosal tissues, which are such common sites for invasion by pathogens and exposure to potentially harmful substances. In addition, many of these ILCs demonstrate significant plasticity. This means their function isn't fixed. It can be altered or skewed by the local environment, particularly by the different signaling molecules, the cytokines, that are present. This allows for a flexible, tailored response, depending on the specific threat encountered.
Let's discuss macrophages and dendritic cells in a bit more detail then, as they're often mentioned as key players in both innate and adaptive responses. Start with macrophages in the alveoli.
Okay, so if foreign material manages to evade the mucociliary escalator and gets down deep into the lower respiratory system, specifically into the alveolar ducts and the alveoli themselves, it's typically phagocytized again, eaten or engulfed by the resident alveolar macrophages. These are fascinating cells. They're actually embryonically derived, meaning they populate the lung during development, and they're self-renewing throughout life. They don't rely heavily on replenishment from bone marrow monocytes like other tissue macrophages often do.
So they're long-term residents.
Exactly. They act as a crucial first line of defense down there, constantly patrolling and cleaning up. They phagocytize inhaled foreign particles, excess surfactant that needs recycling, and cellular debris from normal turnover or injury. They destroy this engulfed material using a potent cocktail of oxygen radicals, destructive enzymes, and halogen derivatives within their internal compartments called lysosomes. And these alveolar macrophages also have another very important role we touched on. They can transport engulfed particles back up to the ciliated regions for elimination via the mucociliary escalator.
Ah, linking the Achilles heel back to the clearance system.
Precisely. It's a slower transport mechanism than the cilia themselves, but it's vital for clearing that non-ciliated zone. Now, if material manages to penetrate deeper into the submucosal areas beneath the epithelium, it encounters recruited macrophages. These are derived from monocytes circulating in the blood. And these recruited macrophages come in roughly two major functional types, although it's more of a spectrum. M1, which are generally pro-inflammatory, and M2, which are more regulatory or anti-inflammatory.
M1 versus M2, what's the difference?
M1 macrophages are the classical angry phagocytic cells. They're activated by things like interferon gamma and bacterial products like LPS. They produce pro-inflammatory cytokines and are geared towards killing intracellular pathogens. M2 macrophages, on the other hand, are often induced by cytokines like IL-4 and IL-13. They can suppress T-cell activity directly or by secreting soluble factors like nitric oxide in mice or immunosuppressive cytokines such as IL-10 and TGF-A. They're more involved in tissue repair and dampening inflammation.
And you mentioned plasticity.
Yes, what's fascinating is the plasticity. For example, under the right cytokine signals, like IL-4 and IL-13, M1 macrophages can actually differentiate or switch towards an M2 phenotype. This adaptability significantly enhances lung defense by allowing the immune system to tailor its response, either mounting a strong inflammatory attack when needed or shifting towards resolution and repair to limit damage. It's a key feature of mucosal defense.
Very dynamic. And dendritic cells, how do they fit into this complex picture? Where are they and what do they do?
Dendritic cells, or DCs, are another critical cell type. Conventional DCs reside mainly in the submucosa, beneath the epithelial layer in the lung, and other mucosal tissues. They act as crucial sentinels. They are considered the major professional antigen-presenting cells, APCs, of the body. Their primary job is to capture and process antigens, pieces of pathogens, or other foreign materials. Once they've captured antigen, they mature, change their shape and surface molecules, and migrate out of the lung tissue to the local draining lymph nodes for the lungs. That's usually the mediastinal nodes in the chest. There, in the lymph node, they present the processed antigen to naive T cells, initiating a full-blown antigen-specific adaptive immune response. They're the link between innate sensing and adaptive immunity.
So they carry the message to the adaptive system. Are there different kinds?
Yes. Besides conventional DCs, there's another important subset called plasmacytoid DCs, PDCs. These function more specifically in an antiviral role. Upon activation, particularly by viral nucleic acids detected via TLRs, they rapidly secrete huge amounts of powerful antiviral type I interferons like IFN or IFN-RA. And both macrophages and DCs, depending on the stimulus they receive, synthesize and secrete a whole range of critical cytokines, those signaling molecules. Things like IL-1, IL-6, IL-12, which drives Th1 responses, TNFO, a major pro-inflammatory player, IL-8, which tracks neutrophils, and IFN-BOOP, important for macrophage activation and antiviral defense. These cytokines orchestrate the entire immune response.
Seems like a complex communication network. Lastly, for the immune cells, let's touch on NK and iNKT cells. What are their specific roles in the lung's defense?
Okay, NK cells, natural killer cells. They originate in the bone marrow like other lymphocytes, but they are a major component of the innate immune system. They are crucial for defending against invading pathogens, particularly certain viruses like herpes viruses, some bacterial infections, and importantly, they also play a role in eliminating tumor cells. They are activated by interferons produced by other cells like PDCs and macrophage-derived cytokines like IL-12, which enhances their killing capabilities.
How do they kill?
Their mechanism of killing is quite direct and doesn't require prior sensitization. They recognize cells that are stressed, infected, or cancerous, often because these target cells lose certain self-markers or express stress ligands. NK cells then release granular enzymes like perforins and granzymes. Perforin literally creates holes or pores in the target cell membrane, allowing granzymes to enter and trigger apoptosis, or programmed cell death. They also synthesize IFN themselves, which further inhibits viral infections and helps stimulate adaptive T cell responses, like CD4 and CD8 T cells. People who lack functional NK cells, for example, are very prone to recurrent, severe herpes virus infections, which really highlights their importance.
Okay, and iNKT cells. They sound similar but different.
Right? Invariant NKT cells, or iNKT cells, they're sometimes considered innate-like lymphocytes, sharing similarities with those T series cells we discussed earlier, particularly in mucosal tissues. They have T cell receptors, but they are semi-invariant, meaning they have limited diversity compared to conventional T cells, and they recognize lipid antigens presented by a special molecule called CD1D rather than peptide antigens. Because of this limited receptor diversity and their ability to respond to lipids, they can respond much faster than classical adaptive immune cells upon encountering their specific antigen. They act as another rapid response force, helping to limit early damage from infection until the more specific adaptive response can mobilize a stronger defense. Upon activation, iNKT cells can secrete a variety of cytokines, including both pro-inflammatory ones like IFNO and regulatory ones like IL-4 and IL-10, helping to shape and balance the overall immune response.
So many layers of defense. Beyond these dedicated immune cells, how do the actual epithelial cells lining the airways, the ones forming the barrier and moving mucus, contribute directly to defense? I'd imagine they're not just passive bystanders.
You're absolutely right. They are remarkably active participants in host defense, far from passive. Airway epithelial cells produce and secrete a variety of antibacterial enzymes directly into the airway lining fluid. Things like lysozyme, which breaks down bacterial cell walls, elastase, and secretory phospholipase A2. These enzymes actively disrupt bacterial structures, leading to cell death. They also synthesize and secrete these fascinating molecules called antimicrobial peptides, AMPs. These are typically amphipathic, meaning they have parts that like water, hydrophilic, and parts that like lipids, hydrophobic, and often carry a positive charge. This allows them to insert into and disrupt negatively charged bacterial membranes.
Like little defensive missiles.
Kind of. For example, beta offensins are synthesized in alveolar type 2 cells, those important cells deep in the air sacs, and they get stored in lamellar bodies along with surfactant, eventually becoming a component of the surfactant layer. Odd-offensins are synthesized and stored within the epithelial cells themselves and also in macrophages and neutrophils, ready to be released upon stimulation. And another group called cathelicidins are secreted from lung type 2 epithelial cells, as well as from neutrophils and macrophages. These different families of AMPs all contribute significantly to killing or inhibiting the growth of a wide range of pathogens, bacteria, fungi, even some viruses, right there at the epithelial surface.
Amazing. It's a chemically hostile environment for invaders. This brings us to another really cutting-edge area, the lung microbiome. We hear so much about the gut microbiome these days, but is there really one in the lungs too, and why has it been so elusive to study?
Yes. While the lungs were historically thought to be a sterile environment below the larynx, it's now quite clear, thanks to modern molecular techniques, that the lung does have a microbiome. It's been difficult to study for a couple of reasons. First, sampling is challenging. Getting samples from the deep lung without contamination from the upper airways, mouth, throat, is tricky.
Right, you have to get past all the microbes up higher.
Exactly. And second, the bacterial density, the sheer number of bacteria, is much, much lower in the healthy lung compared to, say, the gut or even the mouth. We're talking orders of magnitude lower biomass. But it's there. And the composition of this ecosystem actually varies considerably along the respiratory tract, from the upper airways down to the alveoli. Research suggests 3 primary bacterial phyla are commonly represented in the healthy lower airways. Bacteroidetes, which include genera like Prevotella and Bacteroides, Firmicutes, with genera like Veillonella, Streptococcus, and Staphylococcus, and Proteobacteria, which includes Pseudomonas, Haemophilus, and Moraxella. It's a subtle but potentially very important ecosystem.
So what's the significance of this lung microbiome for our health? How does it impact common lung conditions?
Well, alterations in the lung microbiome shifts in its composition or diversity, often called dysbiosis, have been shown to be associated with several major pulmonary conditions. This includes asthma, cystic fibrosis, COPD, chronic obstructive pulmonary disease, and even outcomes after lung transplantation. For instance, the increasing rate of asthma observed in children in developed countries over recent decades has been linked in part to factors that affect the microbiome, like increased antibiotic use, C-sections, and changes in diet.
How does the microbiome link to asthma specifically?
Specifically, studies have found that certain bacterial profiles correlate with asthma. For example, an overrepresentation of organisms in the Proteobacteria and Firmicutes phyla has been linked to asthma diagnosis and severity. There was a really interesting experimental study in mice where researchers showed a significant improvement in asthma-like airway inflammation when the dominant Proteobacteria organisms in the mouse lung were replaced with Bacteroidetes. And this improvement correlated directly with an increase in regulatory T cells, TREGs, and suppression of the Th2 pro-inflammatory cytokines that are prominent drivers of asthma's pathogenesis.
Wow, so the type of bacteria present can directly influence the immune response in the lung.
It strongly suggests that yes, it shows a clear link between the balance of these microbes and our local immune responses.
So could probiotics beneficial bacteria potentially have a role here, just like we see emerging for gut health? Is that a promising area for lung conditions?
That is indeed a very promising, though still early, area of research. Probiotics have shown some utility in treating certain gut infections, like C. difficile, and preliminary studies looking at their use in patients with chronic lung infections, such as in cystic fibrosis, or even for preventing upper respiratory tract infections, have shown some promise, although more rigorous trials are needed.
How might they work?
The exact mechanisms are still being actively researched and likely vary considerably depending on the specific probiotic microorganism being used. However, some studies have shown, for example, that certain bacteria, like strains of Bifidobacterium, can activate dendritic cells and those toll-like receptors, TLRs we talked about earlier, in a way that promotes the secretion of regulatory cytokines like IL-10 and also retinoic acid. These mediators are known to be capable of inducing the development of those regulatory T cells, TREGs, that can effectively downregulate excessive inflammatory responses, like the Th2 response often implicated in allergic conditions like asthma.
Modulating the immune system via bacteria. Fascinating potential. Speaking of pathogens that really challenge the lungs' defenses, let's connect all this to what's been on everyone's mind for the past few years. Coronaviruses, specifically SARS-CoV-2. How do these viruses exploit or overwhelm the lungs' defenses?
Right, coronaviruses. They're a large family of RNA viruses, and they get their name from those distinctive crown-like spike proteins on their surface. While many coronaviruses cause relatively mild, common cold symptoms, 3 have emerged in the 21st century causing severe disease and major outbreaks. SARS-CoV-1, the original SARS, MERS-CoV, Middle East Respiratory Syndrome, and most significantly, SARS-CoV-2, the virus responsible for the COVID-19 pandemic.
And SARS-CoV-2 spread so effectively.
Yes, it's primarily spread by respiratory droplets produced when an infected person coughs, sneezes, talks, or breathes. And crucially, it can be transmitted even days before symptoms appear, or by people who remain asymptomatic, making containment incredibly challenging. Once inhaled, the SARS-CoV-2 virus uses its spike protein to bind to a specific receptor on human cells called ACE2, angiotensin-converting enzyme 2. And this ACE2 receptor is found quite abundantly on the surface of alveolar type 2 cells, those important surfactant-producing cells deep in your lungs.
So it targets a critical cell type right in the gas exchange region.
Precisely. This binding and subsequent viral entry and replication can lead to widespread diffuse alveolar damage, DAD. In severe cases, this can progress rapidly to acute respiratory distress syndrome, or ARDS, which is characterized by severe hypoxemia, low blood oxygen, and respiratory failure, and, tragically, can lead to death. This alveolar damage severely impairs gas diffusion across the alveolar capillary membrane. It also causes capillary leak, allowing fluid and proteins to flood into the air sacs, and disrupts the surfactant system, which normally keeps the alveoli open and prevents them from collapsing. All these factors contribute to decreased lung compliance. The lungs become stiff and hard to inflate, and significantly increased work of breathing. Patients struggle to get enough oxygen.
And what about the infamous cytokine storm? How does that fit into the picture with severe COVID-19?
Yes, the cytokine storm. In some patients, particularly those with severe disease, SARS-CoV-2 infection can trigger an acute, dysregulated hyperinflammatory response. This is characterized by massively elevated levels of circulating pro-inflammatory cytokines, signals like IL-1, IL-2, IL-6, TNF-alpha, interferon gamma, and others. It's like the immune system goes into overdrive and loses control.
And the storm causes damage beyond the lungs.
Absolutely. This cytokine storm can significantly worsen the respiratory distress, but it also leads to severe extrapulmonary manifestations problems outside the lungs. This includes things like dangerous blood clotting events, thromboembolism, leading to stroke or myocardial infarction, heart attack, acute kidney injury, and widespread inflammation of the vascular endothelium, the lining of blood vessels, impacting organs throughout the body. It becomes a systemic disease.
A devastating cascade. Another major way the mucosal immune system can, let's say, go awry is through allergic responses, like asthma. This is so common, but how does the lungs' normally protective defense system get tricked into reacting so strongly to harmless things like pollen or dust mites?
That's a great question. Allergic conditions like asthma and allergic rhinitis, hay fever, are indeed incredibly common, often triggered by ubiquitous environmental allergens, things like house dust mites, proteins from cockroaches, cat dander, mold spores, pollens, or even certain medications like penicillin in susceptible individuals. In a typical healthy mucosal immune response, IgA is the predominant antibody produced locally, focused on neutralization and clearance without much inflammation, as we discussed.
Right, the calm approach.
Exactly. But in an allergic response, there's a crucial switch. Immunoglobulin E, or IgE, becomes the dominant antibody produced against that specific allergen. This switch in antibody class, from potentially IgA or IgG towards IgE, is primarily induced by a specific signaling molecule, a cytokine called interleukin-4, ISO4. IL-4 is typically produced by a subset of helper T cells called Th2 cells. When these Th2 cells get activated by the allergen, they release IL-4, which then instructs the antibody-producing B cells to switch their production specifically to IgE antibodies targeted against that allergen.
So the immune system gets skewed towards making the allergy antibody, IgE.
Precisely. The balance shifts.
And what happens next once this allergen-specific IgE is floating around? What's a chain reaction that leads to asthma symptoms like wheezing and coughing?
Okay, so this newly produced allergen-specific IgE then acts like a loaded tripwire. It circulates and binds with very high affinity to specific receptors called EKII receptors, which are densely expressed on the surface of mast cells, which reside in tissues, including the airway walls, and basophils, which circulate in the blood.
So it arms these mast cells and basophils.
Exactly. It sensitizes them. Then, when the individual is re-exposed to that specific allergen, they breathe in the pollen or dust mite allergen again. The allergen binds to and cross-links these IgE molecules that are already sitting on the surface of the mast cells and basophils. This cross-linking event triggers rapid intracellular signaling pathways within those cells. And this leads almost instantaneously to what's called degranulation, the release of preformed inflammatory mediators stored in granules within the cells, and also the rapid synthesis and release of newly formed mediators.
What kind of mediators are released?
A whole cocktail of potent Th2-associated mediators. These include histamine, a major player in itching, swelling, and smooth muscle contraction, heparin, various prostaglandins and leukotrienes, which are powerful bronchoconstrictors and inflammatory signals, cytokines like IL-4, IL-5, which recruits eosinophils, and IL-13 involved in mucus production and remodeling, and various proteases, enzymes that can damage tissue. It's a rapid and potent inflammatory burst.
And these mediators cause the immediate symptoms.
Yes. These mediators almost immediately cause the classical signs and symptoms of an asthma attack. Contraction of the smooth muscle surrounding the airways, known as bronchoconstriction, which narrows the airways and causes wheezing and shortness of breath, increased vascular permeability leading to swelling, increased mucus secretion, and importantly, the recruitment and activation of other inflammatory cells, particularly eosinophils, leading to further inflammation. So symptoms like wheezing, coughing, chest tightness, and shortness of breath typically appear within minutes of allergen exposure. That's the immediate or early phase allergic reaction. This is often followed hours later by a late-phase reaction characterized by a sustained influx of inflammatory cells, especially eosinophils, leading to more persistent airway inflammation and hyperresponsiveness.
So what does this all mean for long-term lung health, especially for people with chronic asthma who have these reactions repeatedly? Can these inflammatory changes become permanent?
That's a critical point. While the acute inflammatory response following an allergen exposure can often resolve, either spontaneously or with appropriate therapy like bronchodilators or corticosteroids, persistent chronic low-grade inflammation, which is often a hallmark of moderate to severe asthma, can lead over time to a process called airway remodeling.
Remodeling sounds significant.
It is. Yeah. Airway remodeling refers to essentially permanent, often irreversible structural changes that occur in the airway walls. This can include things like thickening of the basement membrane beneath the epithelium, submucosal fibrosis, scar tissue deposition, hypertrophy and hyperplasia of the airway smooth muscle, meaning the muscle layer gets thicker and has more cells, and increased numbers of mucus glands and goblet cells.
So the airway structure itself changes?
Exactly. These structural changes contribute to fixed airflow obstruction, increased airway hyperresponsiveness, and potentially a reduced response to some therapies over the long term. While the exact mechanisms driving remodeling are complex and not fully understood, various cytokines and growth factors released during chronic inflammation, like transforming growth factor beta, TGFF, which is a potent profibrotic cytokine, are thought to play important roles. It's really a striking example of how a highly effective defense system, the immune system, can be inadvertently tricked or dysregulated into mounting a powerful, sustained response against a normally innocuous substance, an allergen, treating it as if it were a dangerous pathogen and initiating its potent inflammatory and repair mechanisms, which ultimately results in the chronic pathology of allergic airway disease and remodeling.
A defense system turned against itself, in a way. Okay, to wrap up this incredible deep dive, let's shift gears slightly to the lungs' often overlooked metabolic side. We've talked defense, but how do our lungs also act as active chemical processing plants, often without us even realizing it?
Right. It's a fascinating and vital function that often gets overshadowed by gas exchange and defense. Beyond those roles, the lungs are incredibly active organs in the metabolism of both xenobiotic compounds, that means foreign substances like drugs, pollutants, or toxins, and also endogenous compounds, meaning substances produced naturally by our own bodies, like hormones or signaling molecules. The key anatomical feature enabling this is the vast surface area of the endothelial cells, the cells lining the massive network of pulmonary capillaries. Think about it. The entire cardiac output flows through this capillary bed with every beat.
A huge surface area exposed to the entire blood supply.
Exactly. This enormous surface area combined with very high blood flow makes the pulmonary endothelium perfectly situated to be crucial for metabolic processing of substances circulating in the blood. They're like a massive, finely tuned chemical filter and factory rolled into one.
Is this metabolic function similar to what the liver does, which we usually think of as the body's main detoxifier? Or are there unique aspects to lung metabolism?
That's a good comparison. While the liver and to some extent, the intestinal tract are indeed the primary sites for the enzymatic metabolism of many xenobiotics, the lungs also selectively participate. They typically have lower overall levels of the major drug-metabolizing enzymes, like the cytochrome P450 family, or CYPs, compared to the liver.
CYPs, those are the enzymes that break down drugs.
Yes, think of CYPs as a large family of tiny molecular machines or chemical factories located within cells, primarily in the endoplasmic reticulum, that specialize in modifying or breaking down a huge variety of compounds, both foreign and endogenous, often making them easier to excrete. While the overall CYP levels might be lower in the lung than the liver, the lungs do express specific CYP isoforms, and sometimes at significant levels. Prominent lung CYPs include CYP1B1, CYP2B6, CYP2E1, CYP2J2, CYP3A5, and notably CYP1A1. This one is found in especially high levels in the lungs of people who smoke cigarettes. Its expression is induced by chemicals in tobacco smoke.
Ah, so the lung adapts its metabolic machinery based on exposure.
Precisely. It demonstrates the lung's specific metabolic adaptations to its unique environmental exposures.
Can you give us some concrete examples of what specific compounds the lungs metabolize, both drugs and natural body substances?
Sure. On the drug side, the lungs enzymatically degrade many medications, particularly those administered directly via inhalation for treating asthma and COPD. This includes inhaled corticosteroids, long-acting beta-2 receptor agonists, LABAs, leukotriene receptor antagonists, and even older drugs like methylxanthines, for example, theophylline. So the lung actively helps process the very medications used to treat lung conditions, which can influence their duration of action and potential side effects. But beyond drugs, the lungs also metabolize a wide array of endogenous substances, things your own body produces that circulate in the blood. This includes vasoactive amines molecules that affect blood vessel tone like serotonin. Also various cytokines, lipid mediators like prostaglandins, and even some proteins. This metabolic processing can occur either inside the endothelial cells, intracellular, or via enzymes anchored to the outside surface of the cells, extracellular. Or the endothelial cells can directly synthesize and secrete important compounds themselves. One really classic and crucial example here involves angiotensin-converting enzyme, or ACE.
ACE inhibitors for blood pressure.
Exactly. That's the target. ACE is an enzyme that is present in very high concentrations on the surface of pulmonary endothelial cells, located within these tiny membrane indentations called caveolae. ACE's job is to catalyze a crucial conversion. It takes a physiologically inactive precursor peptide called angiotensin I, which is circulating in the blood, and cleaves off two amino acids to transform it into the highly active angiotensin II. Angiotensin II is a potent vasoconstrictor. It narrows blood vessels throughout the body, which raises blood pressure. It also stimulates aldosterone release, which causes salt and water retention. This conversion of the pulmonary capillary bed is a major mechanism for maintaining systemic angiotensin II levels and thus regulating blood pressure. That's why ACE inhibitors, which block this enzyme in the lung endothelium, are such effective drugs for managing hypertension and heart failure.
Wow, so blood pressure regulation is happening significantly right there in the lungs.
Absolutely. It's a prime example of the lung's systemic metabolic role.
And what about some other substances? You mentioned serotonin earlier.
Right, serotonin, also known as 5-hydroxytryptamine or 5-HT, is another important example. It's a potent vasoconstrictor, particularly in the pulmonary circulation, and it's also involved in platelet aggregation and neurotransmission. When serotonin passes through the lungs and the bloodstream, it binds to specific transporter proteins on the surface of the pulmonary endothelial cells. It's then rapidly internalized and metabolized by enzymes within the endothelial cell, primarily monoamine oxidase, MAO. This process is incredibly efficient. Approximately 80% of the serotonin entering the pulmonary circulation is metabolized and inactivated in just a single pass through the pulmonary capillary bed.
80% in one go.
Yeah, it highlights the lung's significant role in clearing and regulating the systemic levels of important vasoactive compounds released from elsewhere, like platelets or the gut. And as I mentioned, pulmonary vascular endothelial cells also actively synthesize and secrete important compounds themselves. These include prostacyclin, PGI2, which is a potent vasodilator and inhibitor of platelet aggregation, acting as a local counterbalance to vasoconstrictors. Also endothelin, which is a powerful vasoconstrictor peptide. Various clotting factors are produced. Nitric oxide, NO, another crucial vasodilator synthesized by endothelial nitric oxide synthase, ENOS, various prostaglandins, and cytokines under certain conditions. Interestingly, one thing they generally lack is the enzyme 5-lipoxygenase, so pulmonary endothelial cells typically cannot synthesize leukotrienes, which are potent inflammatory mediators often produced by immune cells like mast cells and eosinophils.
So they make some things but not others. Are there compounds that specifically pass through the lungs without being metabolized?
Yes, that's also important. The metabolic machinery is selective. Some vasoactive compounds pass through the pulmonary capillary bed largely untouched. Key examples include epinephrine, adrenaline, and norepinephrine, or adrenaline to a large extent, dopamine, histamine, the synthetic beta agonist isoproterenol, angiotensin II itself, it's produced there but not significantly degraded there, and substance P, a neuropeptide. This selective processing allows the lungs to clear or activate certain crucial molecules while allowing others, like stress hormones or systemic neurotransmitters, to pass through and exert their effects elsewhere in the body. It's a finely tuned system.
So the lungs can even activate drugs, not just break them down. That's quite a twist.
Yes, absolutely. The lungs play a key role in the metabolism and activation of many prodrugs. A prodrug is essentially an inactive or less active compound that is designed to be converted into its active pharmacological form only after it's administered, often by enzymes at or near the target site.
Why use a prodrug?
There can be several advantages. Sometimes it improves absorption or distribution or reduces premature breakdown. In the context of inhaled drugs for lung disease, activating the drug within the lung can be a major advantage. It can improve the therapeutic index by concentrating the active drug right where it's needed in the airways, while minimizing the amount of active drug that gets absorbed into the systemic circulation and potentially causes side effects elsewhere in the body.
Makes sense. Can you give an example?
A prime example is beclomethasone dipropionate. This is a commonly inhaled corticosteroid used to treat asthma and COPD. As administered, it's a dipropionate ester, which has some activity but is primarily a prodrug. It's rapidly activated by esterase enzymes that cleave ester bonds, which are abundant within the lung tissue itself. This enzymatic cleavage converts it into its highly active metabolite, 17-beclomethasone monopropionate, 17-BMP, right there in the airways where the anti-inflammatory effect is desired. Any dipropionate that might be swallowed is also subject to first-pass metabolism in the liver, further reducing systemic exposure.
Very clever drug design leveraging the lung's own metabolism. What an incredible deep dive this has been into the lung's unsung heroes. From that rhythmic dance of cilia diligently sweeping away invaders, to the intricate chemical and cellular warfare waged by specialized immune cells. And even its often forgotten role as a metabolic powerhouse, processing drugs and hormones circulating through our entire body. It truly makes you appreciate the astonishing complexity humming away within each and every breath we take.
It really does. And if we try to connect this all back to the bigger picture, I think it highlights something fundamental about our body's defenses. It's not just about fighting off an infection once it's already established, but rather about this constant dynamic interplay of physical barriers, like the mucociliary system, and highly specialized cellular and molecular responses, all designed to prevent invasion in the first place, maintain tissue equilibrium, and handle challenges often without us even realizing anything is happening. The sheer sophistication, especially of the mucosal immune system's ability to discriminate and respond proportionately, is truly humbling.
So what does this all mean for you, listening in? Well, next time you take a breath, maybe pause and consider the extraordinary multi-layered defense and metabolic factory working tirelessly, silently within your chest. It also raises an important, maybe slightly provocative question for the future, doesn't it? Knowing now just how exquisitely balanced these intricate systems are. The microbiome, the immune cells, the metabolic enzymes. What might be the long-term, perhaps unforeseen implications for all the novel environmental exposures, the pollutants, the nanoparticles, or even the new therapeutic interventions that we intentionally or unintentionally introduce into this delicate internal world.
That's exactly right. The more we understand these intricate systems, how they work, how they interact, how they adapt, the better equipped we are to appreciate not only the remarkable resilience, but also the potential vulnerabilities of this truly vital organ. It underscores the need for continued research and a cautious approach to perturbing such a complex system.